Here we are in Cambridge at NIAB for the final dissemination meeting of the Legume Plus project. This project's been running for 4 years now and involves many different organisations from across Europe. We've got 2 days of presentations and seminars describing animal health, animal nutrition, environmental impact, and also the agronomy of crops such as sainfoin, red clover and bird's foot trefoil. And this is where we actually get the results that we can disseminate to the wider world.
I talk to hundreds of farmers every year, and I always get a sense of farmers wanting innovation and looking for new ways of producing food in a healthy and commercial way. And I think that products like sainfoin are the key to that future.
Sainfoin did so well in mixtures with different grasses. We did not expect sainfoin to be that powerful in these mixtures.
Cows, when they received 30% of sainfoin in the diet, their intake was excellent actually and was numerically better than in control-fed animals. Growing sainfoin is a good scope for farmers to get sufficient dry matter for their animals and for reducing methane.
According to our experiment, the good concentration is between 25 and 50% of bioactive forage. And for me, it's totally feasible in the field to reach this proportion.
We did actually find a very nice anti-parasitic effect of feeding the sainfoin to cattle. We did actually confirm that you could get an effect on the stomach worms in young cattle.
That we are beginning to get a handle now on what makes some tannins more effective against parasites than other tannins.
We were able to find the structural features that are responsible for the bioactivities we find with the allergy tannins that we utilized in the study. Now we are able to use the information to predict bioactivities of other similar tannins just by looking at the structures as such, and that's really exciting.
Before using the sainfoin against a parasite, you need to characterize the resource. The beauty of these studies is that tannins are everywhere in the world. Once we will have understand the mechanism of action, we have a huge possibility of application.
And luckily we have now tools to understand how the activity is triggered by these very specific structures and properties of the tannins.
There is some potential in it to alter with these condensed tannins, which are actually there in the plant, to have a beneficial effect on the quality of the meat and the milk, which are quite relevant for human nutrition, which then is positive also for the consumer.
It's been a fascinating few days here at the Sophie Taylor building at NIAB in Cambridge. We've been exposed to some brilliant talks, not only by the supervisors, but also the researchers who have been employed by this project and who have formed the basis for the research and actually carried out most of the work that's taken place here.
Without the students, we wouldn't be where we are now, because their enthusiasm and their motivation and their energy to drive this whole field forward has been inspiring and tremendous.
I think that probably the greatest impact of my work in the project has been to produce the tools that also other laboratories can be used to evaluate bioactivity. And personally it has also been very interesting to learn so many new things as a chemist from the other areas.
The genome that we have been able to sequence that we can use in future breeding programs, this is providing a lot of new resource that were not available before.
Since so little time and effort has been put into improving this crop, to make such important steps forward, understanding where the genes are that control the biochemistry and aspects of the way in which it grows, we now have the potential building blocks at least to try and start a proper pre-breeding program.
Breeding forage crops generally takes a long time. From all we learned about the bioactivity of the compounds, I don't think it will be feasible to have an overall variety, but we need to focus on several different aspects for different agronomic environments.
After the results we got during this project, we are quite optimistic. In very intensive production, white clover and red clover will be the main legumes in future, but in a bit less intensive system, sainfoin will have its place, I'm sure.
It's confirmed what we really knew about sainfoin all along. It is a really good forage. And I think what it's also confirmed further than that is that we need to look particularly at the nutritional qualities of sainfoin and the environmental qualities of sainfoin.
It can actually be readily used by farmers, and they can make paddocks with sainfoin, or they can feed pelleted sainfoin, or they can find a related crop.
Farmers are starting to take up some of what we've been working on, and I can see that the project is having impact because people are now beginning to use it, they're beginning to make a business out of it, and animal owners are using sainfoin.
The greatest impact is probably still to come. The main thing would be to utilize our information, which is now very clear, to understand the properties and activities of other types of tannins as well. So that's the main impact that will then come in the future, I think.
The most important outcome of this project is the confidence that it's giving those of us in the industry to utilise legumes to their full, because they are such powerful plants that really do offer us an alternative to what some say is an unsustainable farming system.
All the results from not only this project but the previous project called Healthy Hay will be made available on the legumeplus.eu website. It's been a long couple of days, but it's been well worth it, and I think it's a fitting end to the time, the effort and the investment that's gone into this fascinating project. We are very happy with the output of the project. We've discovered some really interesting facts about the crops involved, sainfoin in particular. If it wasn't for all the supervisors and the researchers and the institutes involved, we wouldn't be where we are today. And it just remains for me to say thank you ever so much for everyone that has contributed to this project. They know who they are, and we thank them for their help, support and their valuable input and the results that they've produced.